So like to thank the organizers for inviting me to talk on aging in the immune system with women-specific aspects. Here are my conflicts of interest. To set the stage, life expectancy gap for people with HIV is narrowing in the modern treatment era, particularly those with a higher nadir CD4 count. It's now close to about 9 years here, and 6 years for those who started ART at a high CD4 count. But a gap in comorbidity-free years persists, even in those with the high CD4 nadirs. About 15 years earlier, people with HIV are getting aging-related complications like chronic liver disease, kidney disease, lung disease, cardiovascular disease, and cancer. And even that gap only narrows to about 10 years in people who start ART at high CD4 counts. So this is a major problem for people getting older with HIV. We think inflammation is a key contributor to this increased risk. It strongly and durably predicts morbidity and mortality in treated HIV infection, including these data from the Insight Network published several years ago. And so our field's goal is really to move people from these top quartiles of inflammation down to these lower quartiles of inflammation. We think people who have low levels of inflammation likely will have a reduced risk of morbidity. But a key question for our field are whether there are sex-based differences in this inflammatory state. Well, we know that women have a stronger innate immune response to HIV than men. In the seminal study from Angela Meyer and Marcus Altfeld done over a decade ago, they showed that when exposed to HIV ex vivo, dendritic cells from men have a much weaker type 1 interferon response to HIV than cells from women. And the mechanistic basis of this difference has now been worked out in more recent years. But the important consideration here is that this may explain why women have lower viral loads than men and higher levels of inflammation at any given viral load in previous studies. And what about those mechanisms? Well, female cells express more of the innate immune sensor toll-like receptor 7. So here we have a male cell on the left. Toll-like receptor 7 is encoded on the X chromosome, so men only get one copy, and they have a moderate amount of TLR7 that gets made. And so when that innate sensor sees viral RNA, you get a moderate type 1 interferon response. But in women, because it's on the X chromosome, they get 2 copies. There's incomplete silencing of one of the X chromosomes in women, such that in many cases, 2 copies of the TLR7 gene get expressed. So you get more TLR7 in female cells, such that when exposed to the same amount of virus, you get a more robust type 1 interferon response. And so this really is the mechanistic basis for higher levels of inflammation in women than men with HIV. And this is borne out in very large studies. This one from the Africos cohort and Hendrik Streek's group, where they saw in a large number of HIV uninfected women in sub-Saharan Africa, a large number of HIV uninfected individuals with HIV negative individuals in sub-Saharan Africa, they saw very little difference in inflammation between men and women without HIV. But among ART-suppressed people with HIV, women had much higher levels of several different inflammatory markers. And several other studies have seen similar findings in recent years. So this TLR7 story likely is a major mediator of this effect. Now, there are also hormonal effects that are important. So we know that estrogen may suppress HIV transcription. In this work by John Carn's group, they showed that estrogen suppresses HIV RNA in vitro, primarily in female cells. So here you see that upon stimulation, you get a lot of HIV RNA produced from infected cells in both female and male participants. But when you add estradiol, in addition to vigorous stimulation, you fully suppress that latency reversal in female cells. Interestingly, you don't suppress it in male cells. So it's not just the hormone, but it's also something about female cells that suppresses HIV in response to estrogen. And what's more, you can see this effect with menopause. So as estrogen levels decline with menopause, you see an increase in the expressible or inducible HIV RNA from cells from women with HIV. So this really suggests that postmenopausal, this may explain why postmenopausal women have higher levels of inflammation in the context of HIV. And this impact on HIV expression is probably even more important in the gut. We know that the active reservoir of HIV in the gut is associated with poor CD4 recovery and microbial translocation during suppressive ART. These are data from our group with colleagues from Merck showing that even in people who have a full recovery of CD4 counts during ART, you have detectable ongoing HIV GAG P24 expression, protein expression in the gut. And that's also associated with CD4 recovery. Those who have poor CD4 recovery have even higher levels of this HIV expression. What's more, the more P24 expression you have in the gut on the x-axis, the more plasma soluble CD14 you have, suggesting a link to microbial translocation. So we think that the increased HIV expression from cells seen in postmenopausal women may actually impact systemic immune activation and possibly microbial translocation in this way. This leads to a theoretical model where pre-menopause you have increased TLR expression resulting in a high innate immune response to HIV, but that's balanced by high estrogen levels, which may have some anti-inflammatory effects, but may also suppress HIV expression. So you have more balanced inflammation premenopausally, but postmenopausally, with declining estrogens, the stronger innate immune response tips the scales and you have increased inflammation. So do sex differences in inflammation have an impact on the risk of morbidity and mortality in treated HIV? Well, our group started to look at this in a CNIX case cohort study that we recently presented at CROI. Briefly, we identified all patients in the multi-center cohort of CNIX who had suppressed their viral load on ART for at least 6 months. There were over 9,000 of them. We took a random sample of them to serve as our control sub-cohort. And then we sampled from all the eligible participants, all the people who got a subsequent vascular event, type 1 and 2 MIs, ischemic stroke, and venous thromboembolism. And then we used Cox proportional hazards models to assess the relationship between inflammatory markers at baseline and subsequent risk of disease. And when we do a large study like this, one of the benefits is that we can start to explore sex differences. And one of the things that jumped out to us from the very beginning was nearly all the inflammatory markers that we assessed were higher in women compared to men. So to the right of one is higher in women. You see all these red bars here showing higher levels in women than men. We wondered whether there could have been a menopause effect, and so the median age is 47 in this cohort. We were actually able to address this and just look at the effect of age on these inflammatory markers in men versus women. And you'll see that in men with increasing age, many inflammatory biomarkers increase. But you see the extent of increases with age is greater in women as compared to men. And this is many of these differences were statistically significant interactions. And so this suggests to us that there is this menopausal effect on inflammation. And there's also a very nice a paper just out in JID from the WISE group showing a very similar thing. What's more, sex may modify the relationship between inflammation and vascular events. And so for type 2 MIs and death, inflammation seems to predict these events more strongly in women than it does in men. Most of these hazard ratios are somewhat higher in women. And a similar pattern we see with ischemic stroke, even though the numbers are quite small, some of these interactions are statistically significant. On the other hand, there may be an interaction in a qualitatively different direction for venous thromboembolism, whereby inflammation seems to predict venous thromboembolism better in men than it does in women, again, with several statistically significant interactions. So this underscores the importance of doing stratified sex-based analyses. And one other example of this is for type 2 diabetes. We saw that while many inflammatory biomarkers predicted incident type 2 diabetes in both men and women, if we hone in on soluble TNF receptor 1, in men, higher levels of this inflammatory marker predict an increased risk of type 2 diabetes. This is exactly what was seen in a much earlier study done by the ACTG and Todd Brown over a decade ago. But because we had a large sample size of women in this study, we were able to actually explore interactions by sex. And in fact, there's a qualitative interaction. In women, higher soluble TNF receptor 1 levels predicted a decreased risk of type 2 diabetes. Now, we don't fully understand the mechanistic basis of this, but this is a very strong difference, which likely represents some really important differences in the biology in the TNF pathway in diabetes between men and women. So lastly, in clinical trials, we need to really pay attention to sex differences. And so our own group is leading a study of the anti-CMV drug letermovir to reduce immune activation in treated HIV infection, because this is another virus that elicits a TLR7 response. We think that this strategy will be even more beneficial in women than in men. And so we're making a concerted effort to enroll women in this study. At least a third women is our target. And that study is just getting off the ground this month, and we look forward to the results in a few years. So to summarize, inflammation persists in people with HIV despite ART and predicts increased risk of age-related morbidity. Women have a more robust inflammatory response to HIV than men, which is likely due to increased TLR7 expression. Estrogen may moderate this inflammatory response and suppress HIV expression in premenopausal women. Postmenopausal women have high levels of HIV expression and inflammation. And lastly, sex-related differences in inflammation may modify the risk of disease in treated HIV, which may impact the optimal interventional targets as well as risk stratification. And so the bottom line is that we do need to recruit more women into clinical trials in these observational studies to better improve the health of older people with HIV. So to close, I'll acknowledge all my collaborators, in addition, Eileen Scully, who generously shared a few slides with me. Thanks very much, and I'll take your questions.